Welcome to our project where we degrade a pollutant called terephthalic acid with bacteria to generate electricity. Firstly, we need to culture a bacteria called Pseudomonas putida so that we have a constant supply of it for our experiments. LB broth is a suitable medium for the growth and reproduction of Pseudomonas putida. LB broth can be prepared with LB powder dissolved in deionized water, and then strongly heating the mixture with constant stirring. We also need to prepare a solution of terephthalic acid, TPA, by dissolving solid TPA in deionized water and strongly heating it with stirring. However, as TPA is just slightly soluble in water, excess TPA powder is filtered out to make a concentrated solution. Both LB broth and TPA, as well as pipette tips used to transfer solutions, are autoclaved to be sanitized. For bacteria culture, a little volume of solution containing the bacteria is extracted and transferred to a new tube using a pipette. Then add LB broth. Parafilm is used to cover the tube to prevent entry of other microorganisms. The original and new tubes are spun constantly. Spinning helps bacteria to be in contact with more oxygen for respiration, and thus promoting growth. Before the experiment begins, we need to centrifuge the bacteria so as to separate it from LB broth. The separated bacteria is then mixed with TPA. The experimental setup features a microbial fuel cell. The anaerobic half cell contains TPA and the bacteria, whereas the aerobic half cell contains distilled water. Carbon rods, which are inert electrodes, conduct electricity. They are immersed in the two half cells and connect to the multimeter through electric wires. A layer of paraffin oil is added to prevent oxygen from reaching the bacteria to ensure it is performing anaerobic respiration. Air is constantly pumped into the aerobic half cell, and agar gel acts as the salt bridge to balance the charges in the two half cells. The electric circuit is then complete and we can obtain results from the multimeter. The reading differs when changing a variable, such as concentration of bacteria, pH value of TPA used, temperature, surface area of carbon rod immersed in TPA, etc. The larger the voltage, the higher the efficiency of electricity generation. This is our project. Thank you for watching.